All right, on the lab. So lab number one, population genetics. Remember when we were talking about the evolution, change in genetic frequencies over generations? And this is the first pre-lab question right there. What is the definition of evolution at the genetic level? Changes in the genetic frequencies from generation to generation. Evolution is the process of changing in genetic frequencies from generation to generation. All right? And this is where we will be talking today about the frequencies, the alleles P and Q and Y is equal 1. I will explain momentarily. So far, so good? All right, next one. What is microevolution? It's a type of evolution which is dealing on the level of population. And today the lab is about the microevolution. Microevolution dealing with the populations. Everything else from the population, species, genus, etc., up into the ranking, we will end up with something which is known as the macroevolution. In the equation P plus Q, see in the equation, math, I know guys, Thursday we're already in math. That's, well, besides the common assignment, that's the only math related lab we will have. So P plus Q equal 1. What P represent? What Q represent? If you read the population genetic intro, so once again, pre-lab supposed to be done before the lab, so try to do it. But basically, two gentlemen, Hardy and Weinberg, come up with the statistical representation or statistical model, the calculation of frequencies of dominant and recessive genes. Remember, most of the genes come in pairs, right? There is a dominant, there is a recessive. They come in two alleles. In bio 1, if you took bio 1, you're supposed to have the example of a regular thumb, which is the dominant trait. And then there is a hitchhiker thumb. Some of you can do that, some of you can't. Put it back, see? Without, some of you can do just that. I can do that, I have that gene. If you cannot, don't worry about it, you don't have that particular allele. Some of you can twist your tongue, kind of U-shape your tongue. I cannot do that, I don't have that particular gene. If you will put your hands together, like this, there will be a particular thumb on top. In my case, my allele, it tells me, my genetics tells me to put left thumb on top. Some of you might have a right thumb on top. If you'll try to interlace your fingers, it's not exactly that, but close enough. If you'll try to interlace the, well, if you're interlacing your fingers without thinking, your trait thumb will be on top. If you'll try to interlace your fingers on the opposite direction, it will not feel natural. You don't have that, technically don't have that allele. Same thing with hands on top. Whenever I'm folding my hands, my left hand on top. If I'll try to do it on the opposite, so my right hand will be on top, it's not feel comfortable for me. You might have it in reverse. Alleles. The genes comes in pairs, right? Okay, and this is what the P and Q stands for. P will be the frequency of dominant allele, while the Q is the frequency of recessive allele. So far, so good? All right, now Y is equal 1. Basically, it's 100%. So if we'll take our population, bio 2 class owners, you will either have a dominant or recessive allele. All of that together is 100%. But instead of saying 100%, they took the 100% divided by 100 and ended up with 1. So we're talking about the decimals. and I'll address that a little bit later today during the lab. Any questions, any problems so far? So P is the frequency of dominant, Q is the frequency of recessive allele. Remember this part, allele, important. Now, let's take a look. In the equation, P plus Q already did question number 3 in the pre-lab. So P represents the dominant, Q represents the recessive. If P is 0.5, all right, 50% of the class have that particular gene, allele of the gene. The other half would have Q, right? So P plus Q have to be equal 1. Therefore, if the P is 0.5, you can automatically find Q. Well, Q will be equal 1 minus whatever the P is. In this particular case, 1 minus 0.5 will be 0.5. So far, so good? Any questions? I can move on. Now, next equation. Now it's a quadratic equation. See? First lecture or first lab, and we're already going into a quadratic equation. So in this particular case, remember the alleles comes in pairs because typically most of the organisms are diploid, right? If they are diploid, you have 2 sets of chromosomes. One comes from the mother, another comes from the father, combination of both. As a result, you might end up with dominant and dominant, dominant and recessive, recessive and recessive. So 3 possibilities. And this is what the next quadratic equation is talking about. P squared plus 2pq plus q squared, nothing else than p squared 2 dominant, q squared 2 recessives, and p squared or 2pq is basically known as the heterozygous individuals. And this is what it means. So P and Q alleles, whenever you start seeing squares, 2pq, q squares, that's the individuals. Now, that's the frequencies of individuals. Before it's talking about the frequency of the alleles, now we start talking about theoretically how many organisms in this particular population supposed to be dominant, homozygous dominant, how many should be hybrids or heterozygous, or how many of them should be homozygous recessive individuals. So far, so good. Please keep in mind that not always the dominant, always the most common allele in the population. Example, human height. I don't really see you guys. Well, I saw some of you via the videos, but I don't really see you. But it's almost safe to assume that pretty much all of you are homozygous recessive for human height. You can say, why is that? Well, there is a dominant trait for height. Those particular individuals, homozygous dominant for the human height, will die during the embryonic development. They will not survive. That's a lethal trait for humans. The hybrids, heterozygous individuals, is something which is known as the little people. They should be under 5 feet tall as the adults, right? And then if you have above normal, and I'm 5'11", so I'm above 5 feet tall, therefore I'm a homozygous recessive for human height. Majority of the populations, majority of human population is homozygous recessive. So in this particular case, recessive is more common than the dominant. All right. Remember, we were talking about the biology, science about the exceptions. So next one. What does it mean that says that population is a Hardy-Weinberg equilibrium? Okay. Here we go, the pre-lab question. What does it mean that to say that the population is a Hardy-Weinberg equilibrium? Equilibrium means nothing changes. The frequencies do not change. Genetic frequencies of P and Q do not change whatsoever. They were 0.5, next generation, and again 0.5. next generation ends again on zero point 0.5, and so on. Make sense? Equilibrium means no change for the frequencies. They remain the same. Now, conditions to disrupt the Hardy-Weinberg equilibrium. There is quite a few of those. Now, get your notes, and you will need to write it down. Why? It will be on the test. It's a short answer on the test. I'm paraphrasing, but it will be something like, name 4 conditions which will disturb the Hardy-Weinberg equilibrium. If I remember correctly, your book gives 7. The lab gives 1, 2, 3, 4, 5. It doesn't really matter. I will not say 5, 7, or 13, or 15. No, I will say some sort of lower number, 3, 4 conditions. So which conditions will disturb the equilibrium? First of all, natural selection. In order for the population to be in the equilibrium, should be no natural selection. As soon as natural selection occurs, the equilibrium will start shifting toward either dominant or recessive trait. Now, no genetic flow. What does it mean? There is no genes coming in from the other population, or there is no genes or individuals which are leaving, taking the genotypes out of the population. That's called the gene flow. There is no mutations. Mutations are random mistakes in DNA, and some of them positive, some of them negative, most of them negative, or something which is known as the silent mutations do not change anything. Out of 10,000 mutations, only 1 will be positive. Excuse me. Under these particular conditions. So far, so good. And then there is a random mating. The mating, in order to maintain the equilibrium, have to be absolutely random. Does that happen in nature? No. There is always a preference, always. And specifically in AMM, well, animals, usually the female will prefer this particular male. Not that one, not that one, not random. There will be something, he's so cute, very nice red fins. I don't like blue fins, I really like red fins. That's my kind of guy. All right, make sense? So, and of course, as soon as we have non-random mating, that's the disturb the equilibrium overall. And then there is the population have to be huge. There is another condition, huge, almost infinite population. If infinite population, then there are all that particular thing happening. If the population is small, then you will, of course, will have all kinds of other things happening. So those particular conditions will disturb the Hardy-Weinberg equilibrium, meaning the frequencies will start happening or changing. If the frequencies are changing, evolution is happening. That's very, very important to understand. Another very important thing to understand, and it will be a second part of the question about Hardy-Weinberg equilibrium on the test. Does any population in the nature, in the real world, yes, huge population, pretty much infinite population. So in this particular case, does any natural populations are in the Hardy-Weinberg equilibrium? And the answer is very loud, resounding no. None of the population in the real world are at the equilibrium. Then you can say, why in the world we're talking about that? Well, just because I decided to do so. No. Why this is important? It gives us an idea. It's our reference point. What do I mean by the reference point? Well, look at me right now. According to my table, according to the video, I'm not moving very fast. I'm moving a little bit within the very confined space, right? From this particular point of reference. I do not move there or there. I'm not walking around. I do not move, technically speaking, from the point of reference of the video camera or webcam. So far, so good? That's a reference point. Now, let's change the reference point. Let's go to International Space Station. I'm moving, according to the International Space Station, I'm moving much faster now. I'm moving because Earth is rotating. Earth is rotating about 1,000 miles an hour. Woohoo! I'm moving with the Earth from the point of view of stationary geostationary satellite. I'm moving really fast. Now I'm moving 1,000 miles an hour. You are moving 1,000 miles an hour as well, from a different point or from a different point of reference. Now let's change that particular reference. Let's kind of look at the reference as the sun. The Earth is rotating around itself, and then it's also rotating around the sun. The speed even increasing even more. Now, if we'll take the reference point, I don't know, the center of the galaxy, all the galaxies right now, all the universe is expanding. We're moving with a whooping, God knows how many kilometers an hour out of the center from the Big Bang, all that particular thing. I don't remember the actual numbers, but we are estimating how fast the universe is well, still expanding. Meaning what? The reference point is very important. How can we tell that population frequencies or frequencies of the genes are happening if we don't have a reference point? So Hardy-Weinberg equilibrium gives us that. It can tell us, we can look it up. Well, we're predicting this in the next generation. And if the prediction didn't happen, then you can say, oops, there is something happening. And this something is evolution. Why, how they changing, that's a totally different problem. But in this particular case, that pure theoretical thing allows us to literally calculate, to see the change in the frequencies of within the population between different generations. Meaning we can predict and see the process of evolution working. Again, how, that's a totally different ball game, but we will be able to today during the lab. It's all pre-lab, by the way, guys, right? So in this particular case, really important is to see if evolution happening or not. Question number 8. How does natural selection affect Hardy-Weinberg equilibrium? We just say that natural selection is one of the conditions which will disturb that. Therefore, it will disturb the equilibrium. Clearly state hypothesis. Hypothesis is supposed to go in if-then format in most of the cases. Not necessarily, but in most of the cases. So let's kind of state something along the line. If natural selection is happening, then Hardy-Weinberg equilibrium will be maintained or disturbed. Maintained or disturbed? Disturbed or maintained?
Disturbed.
Disturbed. You are absolutely correct. Thank you. And yes, those conditions are listed in the lab. It's right above the pre-lab questions. And it's also later on, I'll show you in the lecture as well. So this is the hypothesis. If you want an opposite hypothesis, you can say if the natural selection happening, then the equilibrium will be maintained. That's fine too. It doesn't really matter if it's right or wrong. Don't have to. Hypothesis can be wrong. That's fine. That's part of the science. Now, let's kind of stay with if the equilibrium, if natural selection happens, then the equilibrium disturbed. However, if you don't want to go with the flow, that's fine with me as well. Science, you have a little bit of leeway right there. So now, question number 9. State what would you predict if your hypothesis is true about the Hardy-Weinberg equilibrium of the population of rabbits after 7 generations if lowercase homozygous recessive bunnies are selected again, meaning the natural selection kills them, kind of. They do not survive. Natural selection kills them. Do you think the frequencies will stay the same or the frequencies will change? That's the general idea. What's your prediction after the 7th generation? All right. Well, and the prediction will be probably if the FF lowercase f bunnies are selected again, the frequencies would be changing. Most likely the frequency of lowercase f's will be decreasing. Most likely. I don't know. Well, I do, but let's pretend I don't. I don't want to put that bias into you. So far, so good? Which one are we on? We are on the page number, I don't know, number 3, 4. Question number 8 and question number 9 now. Now, what procedure? Here we have 2 bunnies, 2 different types. The furry bunny, yay, cute one. And then there is the ugly cute, known as the furless. Yes, why they come up with that? It looks strange, really, really strange. After a while, you can get same as anybody seen the, maybe somebody have the hairless cats. And look, first I'm like, hey, something happened to that cat. And then I have a friend, and I came back home in Russia. And I went to, whenever I was visiting him last time, I'm like, Alex, did you got a rat? He's like, it's not a rat. I'm like, what is it? He's like, it's a cat. I'm like, it's running around there. I'm like, what's wrong with the cat? And he's like, it's a hairless. I'm like, really? And then that particular creature comes in, and you're like looking at it, and then it starts doing all the cat things, jumping on the hands, on your lap, start meowing, purring, all kinds of good things. So in this particular case, what we have a population of bunnies hopping very happily over, and some of them are naked, some of them furred, some of them hairless, some of them furry. Everything is good. So far, so good? Everybody's happy?
Yes, sir.
Yes. For the genetic frequencies, well, if the natural selection is selecting, the genetic frequencies will change. Most likely, the lowercase f will decrease. The frequency of lowercase f will decrease because it's selected against, meaning the individuals with that particular genotype will be, those particular individuals will start dying off more often than the regular furry ones. So far, so good. And again, guys, I'm not grading the labs extremely. I'm not a very, whenever I'm grading, if I'll see that you're going in the right direction, I most likely will give you at least a partial point. So it's not extremely, I'm not extremely picky. I'm trying not to be extremely picky, especially in this online environment. So let's take a look on the phenotype. Bunnies, genotypes and phenotypes. So that's the page. I should have, I thought I put the numbers on the page. But in this particular case, capital F and capital F. So homozygous dominant would be furry. All right. Now, capital F and lowercase f. That's the heterozygous bunny, but because it has a capital F, it still be furry. Phenotype is how it look like. So this particular bunny could be either homozygous dominant or could be heterozygous. Unless we're doing some sort of cross test, we cannot tell exactly what his or her genotype. Now on this guy, we can tell right away because it's a furless, it's a lowercase f. So far, so good. It's a homozygous recessive. Any questions on that? Moving on. So in the lab, in the actual face-to-face lab, what I will do, I will give you the cards randomly, 2 cards, and one of them will stay capital F, another will stay capital lower F. I don't know which one. I mean, I'll shift, well, I will shuffle them and I will give you a random card. So whatever your starting genotype is, it doesn't really matter. You can put it on your own. You want to be capital F, that's fine. You want to be capital F, lowercase f, that's fine. Lowercase f's, that's fine as well. Anything you can put right there, I don't really care. I do, as long as you put something something appropriate to this particular class. Now, once again, in the face-to-face lab, what we will have to do, we'll take those particular cards and we will record who has how many capital F individuals we have, capital F lowercase f individuals we have, and lowercase f individuals we have. We'll tie it up to a total number of individuals. In your particular case, it will be 19 because you will be a bunny, basically, during that particular examples, and then we'll calculate the number of capital F alleles in the class, capital lowercase alleles in the class, total number of alleles, and then we'll calculate the frequencies of the capital F, which will be a P, and the frequency of lowercase f, which will be a Q. So far, so good. Unfortunately, I cannot have that particular, I cannot give you the cards, virtual ones, and say, hey, let's kind of do that. So I did it for you already. Yay, thank you, you're welcome. So let's kind of take a look what we have. Our initial lab procedure, within the initial lab procedure, we ended up with 12 individuals having capital F, capital F. It's actual data from one of my classes last semester when we had it face-to-face, so a year ago. Then we had 10 individuals capital F, lowercase f, and 8 individuals lowercase f, lowercase f. So we had 22 furry bunnies and 8 naked bunnies, furless. Total number of individuals within that particular population is 30. Any questions, any problems on that? That's basically just the beginning of the experiment. Now, this is where it gets a little bit confusing, guys. Just a little bit. Total number of capital F alleles is 34. How? All right, look at those particular individuals, right? That one, that particular column, unfortunately, yeah. There is 12 individuals. Each one of them has 2 capital Fs. So 2 times 12 is 24, right? Everybody agree with me on that one? Yeah. And then we have those guys which also carries capital F, but there is only 10 of them. So 24 plus 10 is 34. Make sure you understand where those numbers came from. Let me kind of write it down. 24 from capital F, oops, and 10 from FF. And that would be going to everybody. Total number of F is 34. If you don't understand where I got 34, please ask me right now. Please, 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 please. I know it might be a little bit like, where did 34 come from? Yes, you're basically counting all of the capital Fs. Precisely. Jesus, good comment. Now we'll do the same thing with the lowercase F alleles. Let's take a look. We have, how many we have? We have 10 from capital F, lowercase f, right? And 8 times 2, so totally 16 from FF, lowercase f. So total will be 10 plus 16, so total lowercase f is 26, right? Everybody see where the numbers came from?
Yes, sir.
All right, okay. Make sure that to stop me, because otherwise I will continue to keep those numbers and we will be coming, well, we'll be changing something later on. As long as you understand where the 34 and 26 come from, everything else is very, very easy. So now, in order to calculate P, P will be equal total number of capital F, right? Because it's a dominant, over total number of alleles. Well, total number of alleles, we have 30 total individuals, right? 30 individuals, each one of them carries 2 sets of genes. Therefore, 30 times 2 is total number of alleles will be 60. Therefore, in order to calculate our P, it will be equal to, we figured out that F, number of F is 34 divided by 60. And I already did the calculation, so P will be equal 0.57. Everybody see where it comes from?
Yes.
Okay,
Yeah.
all right. Same thing we'll do for the Q. Q will be equal to total number of lowercase f alleles over total number of alleles. And in this particular case, we calculated that our Q will be equal to lower number of lowercase f's is 26. And it's divided by still 60 because we still have the same population. And then that would be Q will be equal now 0.43. I'm rounding it up. Very important step. Stop right now. Remember our equation, and our equation was P plus Q equal 1. Let's double check that I did my math correctly, so I'm not lying to you. Or in reverse, whenever you will be doing the Florida Panther project, you're not mistakenly giving me the wrong answers. Will take you 5 seconds to double check P 0.57 plus 0.43. Does it indeed equal 1? Not 0.1, 1. Because P plus Q, P is 0.57 plus Q 0.43 have to be equal 1 according to the Hardy-Weinberg equation. Does it? Yes, we did our math correctly. Yes, please. Well, at least this is what I did. So let's kind of go to the 100 easier than that. So this is our initial population. We have 30 bunnies hopping, everything is fine. They're occupying a particular territory. And the very next thing, they will do what the bunny really, really good at doing. Actually, all of the living creatures are really good at doing this. They'll start reproducing. In a lab, in actual lab, what will happen that, remember, we are keeping kind of equilibrium, right? So random mating. So in the lab, I will ask the students to go to random partner, hand 2 cards, the partner will take one card and the student will take another card. So they exchange the genes. Make sense? And of course, it's happening random. So instead of showing, oh, I have two capital Fs, do you want one or another? They are keeping it close. So random mating, so to speak. So far, so good. All right. And in this particular case, after the first generation, what we ended up with is that particular numbers. We had 10 capital F individuals. Did it change from the first initial generation? Yes, it did. We have 14 hybrids. We have only 6 lowercase individuals, the hairless bunnies. So we definitely can see the change phenotypically. We had 22 third bunnies here and 8 furless bunnies in the initial generation. Now we have 24 third and only 6 hairless. Did it change? Yes, phenotypically, yes, but let's take a look. Number of individuals is the same. Total number of alleles, if you calculate 20 plus 14, still 34. 14 plus 12, still 26. The frequency of capital F did not change. The frequency of lowercase f did not change whatsoever. What we can say that this particular population is most likely is in a Hardy-Weinberg equilibrium. But maybe it was a fluke. So I'll say to the students, go again, walk around, meet new people. It's a really good ice-breaking activities. You're reproducing on the very first day, right? And in this particular case, exchange again. And the students will go and they'll exchange the genes again, creating the next generation. And in the next generation, 14, 6, 10, phenotypically. So we have 20 and 10. Very different. Seems like very different. But still 30 individuals, they breed to the capacity. But number of F alleles, capital or lowercase, did not change. The frequencies are stay exactly the same. Is it in equilibrium? Yes. Does the evolution happen? No. Is it probable? Well, is it possible in the real world? No, it doesn't. But it's a really good way to show you how or why we're using the Hardy-Weinberg equilibrium to show the changes in the population momentarily. Once again, maybe that was another fluke, so let's kind of do it one more time. So students go again, exchanging genes, rabbits hopping. So in this particular case, hopping, humping, whatever. So in this particular case, we have 8 of capital F individuals, 18 of hybrids, and only 4 lowercase individuals. Changed. Well, whenever, once again, when we're looking at the frequencies, they stay the same. So we clearly see the population in stasis, opposite of change, right? In equilibrium, the frequency of P and Q stays the same. Does evolution happens here? Yes, no, maybe. A little bit. No, it does not, because remember, the definition for evolution, changing genetic frequencies from generation to generation. It's a process of changing of genetic frequencies. That did it change from generation to generation? Not yet. So far, so good? Any questions, any problems? All right, then. I'm moving on to the next very important part. Again, I already told you I'm Russian, so we're taking that population and we're sending them to Siberia. I talked to my cousin. She's in kind of central part of Siberia. She lives there for whatever reason. But anyway, they had -40. You can say -40 of what? a funny thing. She sent me actually the pictures. I'm like, but negative 40 Celsius and negative 40 Fahrenheit is exactly the same value. That's the only temperature which is cross-secting Celsius and Fahrenheit. Freakishly cold. So anyway, global cooling, not global warming, global cooling happened in this particular population. What will happen to the hairless bunnies? They'll kind of freeze to death. Yes, sorry. Yeah. But again, it don't have to be that. It might be a global cooling or global warming. Then the hairless bunny will be hopping really nicely, and the third ones will start dying from a heat shock. But since I'm Russian and I'm sending you to Siberia, and in Siberia, what happens? Those particular individuals will die off. We still have 30 bunnies, so the rest of them will continue to reproduce to the carrying capacity now. See, Siberia, send the Siberia. Not necessarily the number of individuals, and that's a good idea. But in this particular case, each population usually grows to the maximum of carrying capacity within the area. So in this particular case, if we'll send them to Siberia, now what did we introduce? Natural selection. The cold will kill the creatures which are not adapted to the cold, hairless ones. So now let's take a look what happened. We have 12 individuals versus 18 and 0 lowercase f's because they died. They could not reproduce. We still have the total number of individuals the same. Look, 30. But now the number of alleles changed rather drastically, right? Why? Because those ones that died off. Did it completely eliminate lowercase f? No, it did not. We still have hybrids. And this is very important. Remember that to get rid of the allele in any kind of population, it will take quite a few generations to completely eliminate it from the from the population. So now let's recalculate our frequencies, which I did, and then become 0.7 and 0.3. Yes, because the change happens because of the natural selection. Remember, natural selection is one of those particular conditions which will disturb the Hardy-Weinberg equilibrium. In this case, for the first 3 generations, we had the equilibrium. The frequencies did not change. The number of individuals, haired or hairless, changed, but the frequencies stayed the same. Now, as soon as we apply natural selection, the hairless start dying off because of the cold. We start seeing shift in the frequencies. As soon as we start seeing shift in the frequencies, we start seeing process of evolution taking place. And this is all the lab is all about. So we'll continue. Maybe it was again a fluke. We continue to do the same thing. With the 5th generation, it ended up being 15 capital F individuals and 15 lowercase f individuals. 0 bunnies because they died off. Still 30. They reproduce to the carrying capacity of the population. Now, number of capital F alleles 45, number of lowercase f alleles is 15. Still total number of 60. Now we are at 75 and 25. Precisely. As long as no disturbance for the equilibrium will apply, it will stay in stasis, in the equilibrium. But again, before the equilibrium, we couldn't say, it gives us a reference point. Remember I was talking about the reference point. We're supposed to have, in this particular population, 0.57, 0.43. As soon as we start seeing in the generation 0.7 and 0.3, we know that evolution is occurring because the values start shifting, changing. As soon as we start seeing changing, we can say evolution happening. And then we can say, well, the evolution is happening because of natural selection. or maybe something else. And we will talk about next lecture. We'll talk about something else, some of other driving forces of evolution. Make sense? Because natural selection disturbing the equilibrium. It start well, you can say that you can be on the positive side or on the negative side. You can say that natural selection start choosing, selecting the furred bunnies, or it start killing the furless bunnies. As soon as it start changing that, as soon as it start, somebody will have a preference. As soon as you'll have a preference, remember natural selection, survival, and reproduction of the fittest. The first furry bunnies in Siberia are more fitted to reproduce. They're more fit to survive the cold, harsh environment of the Siberia. As a result, they're most likely to reproduce. But once again, technically that can be changed. After the Siberia, after the global cooling, it could be a global warming. Now the furless will have the upper hand. Make sense? And this is why that particular lowercase f is still lingering in the population. It decreased rather drastically, almost twice as it was before, but still there. In a vain hope that the planet will change, environment will change, and then they will shine. And just checking if there is any other questions. All right, next generation. Once again, we're looking at 1911. Those ones are dying off before reproducing, so we are eliminating them. Still growing to the carrying capacity, 4911 over 60.82.18. And then last one, here we have 22, 8, 30, 52, 8, 60, so 86 and 14. So we can clearly see as soon as the natural selection has been applied, the frequencies are changing. As a matter of fact, guys, you're giving me a great idea. I will not introduce it this time, but maybe next time from Siberia we'll go to Sahara. We'll introduce the global warming in the generation 8, and then we'll see what exactly will happen with the frequencies there. But later on, not right now. So in this particular case, and by the way, Sahara, do you know what Sahara means? I think it's an Arabic word, and if you translate it, it means a desert. So whenever you're saying Sahara Desert, essentially you're saying desert. Anyway, kind of curious fact right there. So in this particular case, we can see as soon as we applied something such as natural selection, we drastically decrease, well, we drastically change the frequencies, and we can actually see the frequencies are changing based on our reference point. That's why Hardy-Weinberg equilibrium is great for the reference point. This is what we're supposed to have. This is what we have in the next generation, meaning that we have some sort of change. If we have a change in the frequencies of genes, genetic frequencies, we have evolution. A micro, small, on the population scale, but we still have it. We're supposed to have 57, we have 86 and 14, 57, 43. Definitely evolution's happening. And now we can say because the environment changed, because of the natural selection. Make sense? Hopefully it does. So lab procedure, how do the frequency in generation 1 through 3, how does the frequency P and Q change significantly during this time? Significantly means more than 5% or 0.05. Did they? No, they did not. Meaning the population is in equilibrium. If it's in equilibrium, there is no evolution. Evolution is change in genetic frequencies. No change in genetic frequencies, no evolution. Ta-da! Now, let me see. It's 11. We are here till 11:45, 35. So we have some time. So in generation 4 through 7, do they change frequencies of P and Q change significantly during this time? Yes, they did. We started with 57, goes to 70, 75, 82, 86. Is the population currently in equilibrium? No, it's not, because the changes in the frequencies. Is this population evolving? Yes, it does. You betcha. It does evolve because we have a change in frequency. Now, the lab will ask you to create some sort of graph. I would really, really recommend you not to do it by hand, but actually use, there is a several things are can be done. The easiest way how I did that is an Excel table. I said plot the graph and I put the numbers and it created the graph, but it's up to you. One of the things which you can do one of the things which you can do if you're not really sure how to do it in Excel or how to create the graph in the Word, it's a really good skill overall. You can print screen that particular picture, print screen it and put it in your table. Right now, I'm giving you the permission to print screen some of the lab parts of the lab, well, of the guide, to put it in your lab. All right? And I'll talk a little bit more about that. Later on, you will have some of the specimens like, I don't know, a shark. And you will have to label the dorsal fin and the tail fin, etc. You can print screen the picture, but I would need the labeling. For this one, I'll give you the permission to print screen it and post it. Now, how you can do there is a several ways how the students doing the labs and submitting it in the PDF. Some of you do it digitally, saving it as the PDF and submitting it. Some people prefer to print it out, and then they have to take pictures. You can take pictures. Most of you have cell phones, and cell phones will use there is several really cool apps which you can use. Let me pull out one really quickly. I'm personally using, and I'm not advertising, I'm personally using Adobe Scan. Adobe Scan is it's a free app from Adobe. You can take as many pictures as you want, and then you convert it, and you can say to convert it in a PDF right now. And it will not be one page per one page per one page. You don't have to submit like 10 pages. It will be one document within 10 pages. I had students before, like page number 1, a picture, page number 2, another file, page number 3, another file. And it takes time, and again, grading is not my favorite part of this particular game, so to speak. All right. All right. And if you have any more questions, shoot me an email. I can go over with that. So let's go with the data. Now we have going to a little bit of math. For example, let's say a given population, 49% of individuals have recessive phenotype. All right. Let me kind of pause it for a moment, just for a moment. Let me get more coffee and I'll be right back. Meanwhile, kind of think if you have any questions, any problems, and we'll go into a data analysis. We'll do some of the little bit more of math. Okay, give me just a moment. All right, back. Usually I don't drink that much coffee, but first lecture always straining my throat. Anyway, so any questions, any problems so far? So that particular chart title you can create on your own, really good idea, or you can just copy and paste, so to speak. Now, let's do some math. And I will be doing it with you, but the main idea is looking what was given to you. Let's say in a given population, 49% of individuals have recessive phenotype. As soon as you will have some sort of number, you will need to figure it out. Is it a P? Is it a Q? Is it a P square, 2PQ, or Q square? Since it says individuals, not alleles, it have to be either P square, 2PQ, or Q square. Now it says recessive. That's our Q square right there. You have been asked to determine what percentage of populations are carriers heterozygous for this trait. You can assume that population is a Hardy-Weinberg equilibrium. So, all right. So in this particular case, what category, what does the genotype of the recessive individuals? The genotype of the recessive individuals would be Q squared, all right? Well, for recessive individuals, technically, I'm sorry, I jumped there. For the recessive individuals, pick a letter, any letter, whatever. Give me the letter. Type a letter which you want. Whatever letter. Type it in the chat box. We'll use that letter. Anybody? Any favorite letters? M. All right. So, good. Thank you, Marisa. So in this particular case, we decide that we will call whatever the trait is, it will be our M. So if it's a recessive phenotype, it has to be lowercase f and lowercase f. M, I'm sorry, M. We used M, F before. So lowercase m and lowercase m. All right, and it would be Q squared. Yeah, I jumped the gun a little bit there. So now, if 0.49, how we figured out that 0.49 is Q squared? Remember that 49%? We took 49% and we divided by 100%. Why did we do that? In order to get rid of the percentages. And if you'll do this very carefully, you will end up with 0.49. 0.49. And therefore, our Q square will be equal to 0.49. Now, if we know that the Q is 0.49, can you find the Q? Well, if Q square is 0.49, we can square root it, right? Then Q, after square rooting it, will be 0.7. Everybody see what I did there? Kind of hard to write the square root. So, and if we know that the Q is 0.7 and we know that P plus Q equal 1, P plus Q equal 1, so, P plus Q equal 1, P plus and Q is 0.7 equal 1. So P will be equal 1 minus 0.7. And after that particular extremely hard Yep, exactly. See, 0.3. Great mind thinks alike. Good job. Yeah, good job. Okay. So any questions on any of those numbers, where they come from, where we'll figure it out. Once again, as soon as you have one, you can logically figure it out another. So if, well, we got what is a P, P is 0.3, what does P represent? The genotype is MM, yes, for the first question. Oh, because Marisa chose the M as the letter. If you don't like M, you can choose whatever you want, as long as it's a lowercase, both of them. All right? So think about it. If you don't, you can actually send a private question in a private chat. I will not tell who from who. But if you are not, well, if you are slightly confused where the numbers come from, it's a really good time to ask. We are doing really well. We probably will be able to finish it a little bit earlier today as well. You are doing awesome, guys. All right. What does P represent? Remember, as soon as we're talking about squares or 2PQ, that's individuals. As soon as we're talking about P or Q by itself, that's the frequency of alleles. P is the dominant allele, Q is the recessive allele, frequency. Not just the recessive allele, no, the frequency of. In this particular case, what does P represent? P represents the frequency of dominant allele. Now what you know, P, calculate P squared and 2PQ. All right, well, let's do that. Your sound cut off. Everybody can hear? Yes, no?
Yes.
Okay, 0.09, exactly. So P square, E square will be equal 0.3, which is our P, and we also squaring it, and that will ended up is 0.09. Now 2PQ, basically exactly the same thing as we had before. 2 is a number, we're multiplying it by P is 0.3, and then we're multiplying it by Q is 0.7, and then it will be 2 times 0.3, 0.6, 0.6 times 0.7, 0.42. In this particular case, it's a really good idea to stop for a second, maybe a little bit more than a second. And remember, P squared plus 2PQ plus Q squared equal 1. Double check your math. Double check my math. So our P squared, so P squared plus 2PQ plus Q squared have to be equal 1. Let's plug the numbers which we just found. Our P squared is 0.09 plus 2PQ is 0.42 plus our Q square was given to us, it was 0.49. Does it indeed equal 1? 0.09 plus 0.42, 0.55, or 51, 0.51 plus 0.49 is 1. Yes, we did our math correctly. Do not skip, I beg you right now, do not skip the double checking point. I'll talk about that a little bit more later when we'll do the assignment. We will start working on a common assignment. Takes you 10-15 seconds to double check, but if it's not equal 1, it means that you did something wrong. Look around and see what you did wrong. Okay? I would highly recommend you to double check. I've seen the students doing 0.3 times 0.3 and getting 0.9, not 0.09, but 0.9, 90%. And then they're taking that number and they're saying my P square is 90%, my PQ, my 2PQ have to be equal 180%, and it's becoming a huge problem. So don't skip that step. I do understand it's an extra step, but it's overall later on might result in a better grade for you. I didn't skip it. I probably by now can remember it by heart, but I still double checking. Sometimes it could be just a little bit, tiny little bitty mathematical issue. All right? Yep. All right. What does P represent? Already talk about that. What percentage of populations are carriers? What are the percentage? So basically carriers or heterozygous without 2PQ. Since it's talking about the percentages, we know that 2PQ is 0.42 times 100%. Well, under that being 42% of that particular population are carrier for the gene M. Well, lowercase gene M. All right. We are on the closer to the end. We in the post lab review questions, and we solved. Oh no, we're not in the post lab. We are still in the lab, and we are going. Alternatively, we can work in the opposite direction as well. In the instance, the entire population has been genetically tested. Hannah, all of that is recording, so it will be available to you, so you can catch up whatever you didn't get. My general speech for the last 5 minutes were make sure to double check. All right, so alternatively we can work, let's say, the frequency of dominant allele. As soon as you have some sort of numbers, you need to figure it out. What is it? Again, P, Q, P squared, 2PQ, Q squared. So in this particular case, it says allele. It has to be either P or Q. It says dominant allele. It has to be a P. So if our P, we just figured it out. If our P is 0.1, can we figure it out Q? Well, yeah, Q will be, so 0.1 plus Q equal 1. Q has to be 0.9. If we have that, well, P squared now will be 0.1 squared, which will be 0.01. Q squared will be 0.9 squared would be 0.81. Now 2PQ. 2PQ will be equal 2 times P is 0.1 times 0.9. 0.2 times 0.9 would be 0.2. 0.2 times 0.9 would be 0.18. So now all I need to do is double check myself. P squared, so 0.01 plus 0.18, my 2PQ, plus 0.81, is it, oh, 81, not 91. 81, is it indeed equal 1? Let's see, 0.1 plus 0.18 will give me 0.19, 0.19 plus 0.81 indeed equal 1. So I double check my math is correct. Calculate Q, we did. Calculate P squared, we did. Q squared did. PQ, we did. Now, give me the answer for this one. What percent of this population do you expect to have the dominant phenotype? Since it's a percentage, give me the percentage. Type it in. Either in an overall chat or private chat, doesn't really matter. I want to see the numbers. Tell me what is percentage of that population do you think is in a dominant phenotype? I see a couple of ones. Anybody else? Again, 10%, okay. 1%, 10%. All right, Hannah. Once again, video will be available to you today afternoon. Let's kind of focus on the thing in the at the present. What percent of this population do you expect to have the dominant phenotype? I have several answers of 1 and I have several answers of 10. 10 I have. Ooh, I have 19 now. Even better. So we have 3 possible answers. 1, 10, 19. Anybody more? Can we make it 20 or 30 or 40 or 50? Overall, good answers. Some of you active, some of you not so much. Again, remember attendance. And the correct answer is 19%. Why 19? Let's read the question again and figure it out. Remember critical thinking? Let's critically think this thing. Dominant phenotype. If it would say dominant genotype or homozygous dominant genotype, then yes, 1% would be correct. I agree with you on that one. But it says dominant phenotype. They look like dominant. So it's your P plus 2PQ. Because 2PQ, the hybrids, they still look dominant. Look, phenotypes, how it looks like. So in this particular case, we have P square is 0.1, 2PQ is 0.18, 0.01 plus 0.18 will be 0.19, and you multiply by 100 in order to get percentage. So it will be 19%. Make sure to read the question, please. Everybody understand where? Okay, 19. Laura, you just asked, do you understand now why it's 19? Why not like 10 or anything like that? Tiny little bitty trick. And yes, some of you got it right away. Some of you didn't look at the terminology, phenotype. Once again, terminology, keywords is extremely important in bio. So be sure before answering the question, understand what the question is asking. Because if you don't know what the question is asking, it's kind of hard to come up with the correct answer. It's basically like I'm saying, I'll tell you right away, give me the answer. I'm like, what the question? I'm like, no. There is no question. Give me the correct answer. Can you give me the correct answer to the question you don't know? I guess, yes, no, maybe, right? So every time you are in any kind of test, and we're talking about specifically in biology, but any test, figure it out what the question is asking. Think a little bit critically, so to speak. If you don't like critical thinking part, and everybody's saying critical thinking, I come up with another term. Actually, I heard that not so long ago. Let me find the note. It was instead of critical thinking, it's called, let me find it. Feedback. It's called Analytical reasoning. Here we go. Analytical reasoning. Analyze what is giving and give me the reason for it. Phenotype you're using calculations if it says dominant, because look, in this case, later on, one of the students asked, so with the phenotype we use calculations p squared plus 2pq for the dominant, because hybrids 2pq and homozygous dominant p squared will look dominant. But whenever you're looking at the recessive phenotype, which will look recessive, like in our particular case, whenever we're talking about rabbits, rabbits were the naked rabbits, the furless. So a furless rabbit, recessive phenotype, that's just a q square. But the haired ones, it could be either p square or 2pq. So in this case, next question, what percentage of the population you expect to have a recessive phenotype? That would be q square, and q square is 81, right? And yes, in this particular case, 81% of whatever you'll see will have a recessive phenotype. Is it possible? Yes. In some cases, recessive phenotype shows up in the population more often than the dominant. So let's take a look. You revisit this population after 20 years to look at the next generation. You notice. So remember, we had 19 and 81. Now you're seeing 36 and 64. Do you expect those particular numbers? I have no idea where the 36 came from. I did not expect it. I have no idea where 64 came from. No, I did not. I did not expect to see those particular numbers. Is this particular population in equilibrium? No, it's not, because again, remember, we had 19 and 81. Now we have 36 and 64. Because they changed, it's definitely not in equilibrium. What some things could happening in this population? Any of those things which are disturb the equilibrium. Natural selection, mutation, non-random mating, immigration or emigration gene flow. We might have a bunch of dominant phenotype coming in, or we might have a bunch of recessive phenotypes leaving, or in this particular generation, let's say the female decided that the dominant genotype, dominant looks good. Let's kind of mate with him and let's drop all of the recessive. We don't know what happened, but we can figure it out. Non-random mating, gene flow, mutations maybe, but usually mutations will not drastically alter that. Might be reduction in the population, a drastic reduction in the population. There was some sort of catastrophe and most of the recessive guys died off. Remember, as we had in Siberia and we had the Siberia and the hairless bunnies. So that's basically it with the lab. And yes, guys, if you have some sort of issues with the connections happens, try not to have that on a regular basis. But yes, videos exactly for that. If you have some sort of problem, then come back and rewatch them. Now, post-review lab questions. Post-review questions, make sure that you will answer them. We'll do some of them together, and then I'll let you go because we are about 5 minutes till the end of the class. So, what was your original hypothesis? We thought that if natural selection occurs, then the equilibrium will be disturbed. Based on your lab data, do you need to accept or reject your hypothesis? Well, if this is the hypothesis you went through, then we are accepting that hypothesis, because as soon as the natural selection started, we start seeing the changes in the frequency. In real rabbit habitat, new animals often come to go, immigrate or emigrate. How this movement can affect the allelic frequency, it will definitely change it. It will not stay the same. So it's one of those particular things which will disturb the equilibrium overall. So number 4, which of the 5 conditions necessary for Hardy-Weinberg equilibrium to be affected? Which of all of them? Well, any of them, actually, no, any of them. So any of those particular things happening, we're talking about natural selection, non-random mating, gene flow, immigration, the mutations. What else was there? Something else. All of those will affect the equilibrium. So make sure that you know those particular conditions. Don't have to be in a specific order. Once again, the question on the test, on the upcoming test, on test number 1, I promise it to you, will be something like at least 3 conditions for the Hardy-Weinberg equation or equilibrium. Does any of the populations in the Hardy-Weinberg equilibrium in real life? So is the population, so let's see, would the population be in equilibrium for number 4? If those conditions happening, it will not be in equilibrium. As soon as it's not equilibrium, the genetic frequencies are changing, therefore it is evolving. How might simulate effect if you repeat this activity? How you might simulate this effect if you were repeat to this activity? We could go with the global, well, instead of global cooling, we could go with the global warming. Send them to Sahara Desert, for example. And then we'll see that the lowercase f, the recessive genes will start prevailing in the population because they have a higher chance to survive and reproduce. List conditions. Okay, so 5, 6. How the result of the simulation as example of evolution? Genetic frequency changes. Genetic frequency changes means we have some sort of process changing them, and that's evolution. List conditions necessary for the Hardy-Weinberg law to be valid, and those are 5, well, according to the lab, 5 conditions. The no natural selection, no mutation, random mating, no immigration or no gene flow, and what was the 5th one? Well, I said that we have, let's see, random mating, no genetic drift. Here we go. Yeah. Genetic drift is a random event which changing the, drastically changing the percentage in the population. We'll talk about that during the lecture next time. What is the evidence of evolution is occurring in any given population? Evidence of, well, change in frequencies of the genetic alleles. Nowadays, we have the means to figure it out the actual genetic alleles in the population and compare it to the next generation, right? All right, so question number 9 as well as the question number 10. Do it yourself by next time, by Thursday. If you'll run into any kind of problems, I will go back or remind me to go back to look at those particular numbers. If you have any questions, any problems on how to do number 9 and number 10, make sure to ask me those particular questions. Those are similar questions you will see on your group project. So I really want for you to figure it out how to do it by yourself. It's not really hard as long as you're going step by step. And don't forget the step of double checking yourself. All right. Now on every lab, lab needs to be uploaded on the Blackboard by Sunday, 11:59 p.m. Anytime between now and then. Yes, I will send the, yes, there will be a video coming in, and then the lab needs to be uploaded by Sunday. On Thursday, I will ask you guys, do you have any questions on the question number 9 and number 10? And if you will have a question, if you're stuck somewhere or anything like that, you can either send me the email or we can go as the class together. The next lab will be shorter than this one. This is the longest lab out of all of them. I will send the announcement about the video and questions today afternoon. I have a question. Man, you guys eager right there. No problem. So now let's take a look. 11, question 11, reflection questions. Every lab will have that particular question. Every lab, and for every lab, you will need to write something down. I would highly recommend you to give me a little bit more, since we are not in a face-to-face environment, so to speak. It gives me the perspective on, did you learn something? Do you have any issues? Do you can you use it somewhere? Now, hopefully, you'll be able to talk a little bit about the evolution. Hopefully, I don't know if you want it. I'm not trying to indoctrinate you into the theory of evolution. You have to believe in evolution, or otherwise you'll fail biotech. No, that's not my part as the professor or anything like that. It's up to you. But my deal is explain the evolution. I have students asking, do you believe in evolution? Well, belief is a religion. Evolution is a scientific fact. I can accept or I can reject that particular scientific fact. And we'll talk about more about the evolution throughout the semester, but specifically next lecture as well. I'll talk about some of the main misconceptions. And am I accepting the evolution? Yes, I am. Do I believe in God? No, yes, pretty much I do. So in this particular case, you can say how you can believe in God and believe in evolution. I don't believe in evolution. Evolution happening. And the Catholic Church at some point of time says, well, the Pope come out and says that yes, evolution happening according to the God's design. And we'll discuss that there is a huge, lots of problems, lots of issues with evolution, and that's one of the reasons why it's still a theory. But going back to question number one, reflection questions. Don't just tell me main purpose of what you did in the lab. In the lab, we did population genetics. Well, give me a little bit more substance. I do understand that it takes a little bit more time for you to write down. Don't have to be a complete sentences, don't have to be a proper spelling. I'm not a huge guy on the spelling, so which is good for the lab practical. What you learned, what did you observe? Give me a little bit. Some of you will do, some of you will try to sneak out of that, and I'm quite often I'm looking, one of the first things in the lab I'm looking at those particular questions. Did you do them? You didn't do them? Did you do them well? And since you are honor class, well, I expect from all of my students to answer those particular questions, but for the honors class specifically, we're going a little bit deeper. So give me your perspective. Maybe you dislike that particular lab completely. Write it down. I hate this lab. I don't know why it's here. So I need a little bit of feedback, and those particular questions very often gives me a very nice insight of what I need to go over in the future, so you will be successful. All right, that's pretty much it for today. Thank you very much, guys, for joining me, and I'll see you Thursday. Well, we are done, so 16 out of 16 weeks, so 32 classes, basically 31 remains. Thank you, guys. I appreciate coming in. It was a good
Thank you.
turnaround. Thanks.